Okay, so chapter 3 is all about cell structure and function, which should be a good review from some previous classes. Now, what this slide shows is basically just a prototypical cell. So you can see that we have a plasma membrane that surrounds the outside of the cell and kind of holds it all together. And we find that there are a lot of intracellular organelles within it who all play a vital role in helping to maintain cellular homeostasis. So we can see things like the nucleus, which is going to house most of our genetic code and have most of our genes, which can help direct the production of certain proteins, which all have important functions in our cell. Now, within the nucleus, we have the nucleolus, which is the site of ribosomal RNA synthesis. Ribosomal RNA, or rRNA, is necessary for ribosomes to help make protein. Now, we can see other organelles like the mitochondria, which makes most of our cellular ATP. We have other cytoskeletal elements like microtubules and actin filaments and maybe some microfilaments. And these are all involved with basically supporting the cell structure and helping with the movement of organelles and material within the cell, as well as moving the entire cell itself. Now, we also see some Golgi apparatus over here in blue. The Golgi is kind of like people talk about as being the packaging and processing center of the cell, where the Golgi can help process proteins to become active or inactive, or kind of change the activity of a certain protein. And the Golgi apparatus can also package those proteins or products into secretory granules, which then can be taken over and secreted out of the cell via exocytosis. Now, we find other organelles like our rough endoplasmic reticulum and our smooth endoplasmic reticulum. Rough ER, or rough endoplasmic reticulum, is involved with making proteins that are secreted. The smooth ER is involved with lipid metabolism, so like making and breaking certain lipids. Now, there are also vesicles in the cell that are involved with different functions. Like we have lysosomes and peroxisomes. Lysosomes are involved with the breakdown of damaged intracellular components. Peroxisomes, think of as like the mobile forms of smooth ER. So these peroxisomes are involved with lipid metabolism and things like that. Now, this is just an overview of organelles here. And what I want you guys to know is basically the major functions of each of these organelles. Now, we'll talk about these a little bit throughout this lecture, but again, I want you guys to know the major functions of each of these organelles. Now, the first sort of organelle we'll talk about is the plasma membrane. And the plasma membrane is this phospholipid bilayer that surrounds the entire cell and kind of holds it all together. And it does this because phospholipids are basically nonpolar, meaning that they are fat-soluble or hydrophobic or water-fearing. So what happens is the phospholipid bilayer, because it's made of molecules that are water-fearing, it repels water. So that water on this side of the membrane and this side of the membrane are repelled and actually prevented from kind of crossing the membrane. So the plasma membrane actually forms a protective water barrier surrounding the outside of your cell. Now, embedded in the plasma membrane, we have lots of different transport proteins which are involved with basically transporting molecules into and outside of the cell. Now, if we look at the plasma membrane in another view, we can see that, we have this phospholipid bilayer out here, and this is looking at a cutaway view at the kind of the sphere of a cell. You can see that the phospholipid bilayer forms a water-filled center, which would be the sort of the intracellular cytoplasm. Now, the reason why it's water-filled here in the center is that water is actually repelled by the membrane here, so that once water gets trapped inside, it's more difficult for it to leave. It's also difficult for water to just enter the cell through the plasma membrane. Now, water can enter the plasma membrane a little bit because it's kind of small, but for the most part, water is repelled by the plasma membrane on either side, whether it's extracellular or intracellular. Something else we find embedded in the plasma membrane are these glycoproteins, which are used for cell recognition. So these glycoproteins we've talked about before as being like the name tags of the cell, or you can think about it like the gang sign as well, where we have these sugar molecules here, and each one has a specific shape, And the shape of these sugar molecules really helps tell other cells and your immune cells that it might be like a cell of your own body, or it can also kind of provide some information about what's going on within the cell. In fact, there are some of these glycoproteins which are markers for cancer, and immune cells can recognize a glycoprotein as being the cancer glycoprotein, and that your immune system can actually start to kill off that cancer cell based on the certain type of glycoprotein that's being expressed in the membrane of that cell. Now, the cytoskeleton is really what holds the cell together, and we have the membrane skeleton, which is associated with the plasma membrane. Now, the membrane skeleton really just helps support the shape and structure of the cell and gives it more of that rounded, spherical appearance. And the actin skeleton is involved with actually cell movement, because some cells can actually move around via like amoeboid movement. The actin skeleton is made of these intermediate filaments or microfilaments that help the cell contract and move around, maybe to help chase a bacterium or to migrate in a tissue during development. Now, we talked about the endoplasmic reticulum earlier as having rough and smooth components. Remember, the rough ER is involved with making proteins that are secreted. Smooth ER is involved with lipid metabolism, so making and breaking lipids for the cell. And what's interesting is that the endoplasmic reticulum is also highly associated with your Golgi apparatus or Golgi body, so that the proteins that are made by your rough endoplasmic reticulum get packaged into vesicles. Those vesicles are transported to the Golgi apparatus, whereby the components in those vesicles, like protein, are processed by the Golgi, and then they're repackaged into vesicles, which are transported over to the plasma membrane, which then can be secreted. An example of this Golgi secretory pathway could be like insulin. So insulin is a type of protein hormone. It's gonna be made by your rough endoplasmic reticulum. Now, once that insulin is packaged into vesicles, it can get transported to the Golgi, whereby enzymes in the Golgi can actually help cut that insulin protein into pieces that are actually more active or ready to be active. That way, once the insulin is secreted, it's going to be secreted in its active form, not the inactive form. So the mitochondria is involved in cellular metabolism, and it's a pretty interesting organelle that has several membrane components within it. Now, what's important to note is that there are these transport proteins you find in the inner mitochondrial membrane that are involved with something called the electron transport chain. Now, basically what happens is that certain substances kind of get fed into this cycle where there are electron carriers that can shuttle electrons over to this electron transport chain, whereby these electrons can be used to pump hydrogen out here into this particular space over here. What this does is creates a concentration gradient where there's more hydrogen ions in this outer mitochondrial space than the inner mitochondrial space. And with this other protein over here called ATP synthase, hydrogen ions can flow down through ATP synthase in the direction of its concentration gradient. And when it does this, it causes this protein to spin really fast. And when this spins really fast, it provides the kinetic energy to fuse ADP and inorganic phosphate to, guess what, make ATP. So the spinning of this ATP synthase due to hydrogen flowing through helps create ATP, which basically is a form of usable energy for the cell. Now, when we talk about cellular metabolism, it's really just biochemical processes where foodstuffs, are basically just components of our food, are used to provide cellular energy and other biomolecules from the cell. Now, we can talk about metabolism as a whole as being all of the chemical reactions that are occurring in all of your cells in your body at any moment in time. But we can divide metabolism into two main components. We have anabolism and catabolism. Anabolism is going to be the form of metabolism where you actually use energy to make larger molecules. Okay, so anabolic reactions will typically result in the synthesis of more complex molecules, like larger proteins or longer chains of sugars. Now, this is different from catabolism, which is actually going to be the breakdown of molecules into smaller components, and this is typically going to be energy releasing, and that energy that's released from the breakdown of molecules can be used to power other cellular mechanisms. Now, when we talk about the type of metabolism that helps us create ATP in our cells, it starts with this process called glycolysis. Now, glycolysis literally means to break down sugar. Glyco is sugar, lysis means to break apart. And glycolysis is an anaerobic process, meaning it does not require oxygen to power this process. Now, glycolysis, in the process of breaking down sugar, can ultimately lead to the production of 2 ATP molecules. It actually makes 4 ATP, but it takes 2 ATP to help break down the sugar, so it yields a net 2 ATP molecules for each glucose that's broken down via glycolysis. This is anaerobic metabolism. Now, the breakdown products of glycolysis can be fed into what's known as the citric acid cycle, or Krebs cycle, and basically the 2 acetyl groups that are broken down from sugar, from glycolysis, they help feed into the citric acid cycle or Krebs cycle to yield more ATP as well as more electron carriers like NADH and FADH2. Now what happens then is that those NADH or FADH2, those are electron carriers that can be shuttled on over to the electron transport chain, whereby they feed into this process called oxidative phosphorylation, where basically electrons are transported down that chain of proteins. Hydrogen ions are pumped into that outer mitochondrial matrix, and then that movement of hydrogen ions back into the inner mitochondrial matrix helps spin ATP synthase, which allows for the fusion of ADP and inorganic phosphate to make ATP. But this requires oxygen because oxygen is the final electron acceptor in this process. And so if you don't have oxygen present, it can't accept those electrons and you can't undergo oxidative phosphorylation. So if you look at this process, this is just outlining glycolysis in general. And you can see that the breakdown of glucose can require some energy input. Because glucose is a fairly stable molecule, it requires some energy to break it apart. And so we have to have 2 ATP used by enzymes that can break down this glucose molecule. Now there's a series of steps that help reduce this glucose molecule into smaller molecules like fructose here. And then that fructose can be split into 2 different pathways, which will ultimately yield something called pyruvate. And you can see in the process of converting fructose into 2 pyruvate molecules, we yield 1, 2, 3, 4 ATP and 2 NADHs. So we can talk about glycolysis as yielding a net 2 ATP and 2 NADHs. Those ATP can be used directly by cellular mechanisms right as glycolysis occurs. Otherwise, those NADH molecules can be transported on over to the mitochondria, and those can be fed into the electron transport chain for oxidative phosphorylation to occur. Now what this is showing is that those acetyl groups that come from glycolysis can feed into this Krebs cycle or citric acid cycle. Now I won't ask you guys to memorize this whole cycle, but what's important to note here is those acetyl groups from glycolysis, they're fed into this cycle whereby there's a long chain of basically enzymatic reactions that help convert molecules into other molecules. And in this process of converting molecules, what we get out of this process are some forms of transportable chemical energy, like NADH, more NADH, FADH2, as well as some ATP. Now, those NADH and FADH2 can be used for the electron transport chain to make even more ATP. Now, what's also important for a cell to undergo are different types of transport. Now, membrane transport for larger molecules is going to require the use of vesicles. And so there's two main processes here for transport of large or macromolecules. We have endocytosis and exocytosis. Endocytosis is a process of taking molecules into the cell by using a vesicle. And there's two subtypes. We have peno and phagocytosis. Penocytosis is just like ingestion of water in small molecules by taking gulps of that extracellular water. And then phagocytosis is an ingestion of larger molecules that typically involves things like receptors that can kind of activate that process. Exocytosis is basically cellular secretion, where cellular products can be packaged into vesicles, transported to the plasma membrane, and then exocytosed out of the cell. Now we can see this process here where this is the plasma membrane, okay? And what we find here then is that if this is the extracellular material, and this is intracellular, what we can do is actually kind of take that material in, and then that forms a vesicle, which then can be transported around within the cell. And this process here would be endocytosis because you're bringing something into the cell. Now what's interesting though is if you look at this going the other way, going this way, that's actually exocytosis, where we can take something in a vesicle, fuse it with the plasma membrane, and then expel some material out of the cell. So we have endocytosis going this way, and then exocytosis going the other way. So they're similar processes, just kind of defined on what the direction of movement is. Now there are other ways to transport molecules across the plasma membrane, and this can involve things like ATP-driven pumps, different carriers, as well as channel proteins. Now, understanding whether or not we need to transport something across the plasma membrane requires our knowledge of, the electrochemical gradient. And this electrochemical gradient is really just a gradient or a difference in the concentration and charge of molecules inside and outside of a cell. Now, this electrochemical gradient can be used to help transport molecules into or out of the cell, really just depending on what the gradient is. Now, there's two main types of transport here. We have passive and active transport. Passive transport is really just facilitated diffusion or just diffusion itself. And passive transport does not require any additional energy input. So it doesn't require ATP to transport molecules. An example of this would be like the transport of water across the plasma membrane. Water just moves via osmosis, which is a passive process. It does not require ATP for that to occur. This differs from active transport, which does require the use of ATP. And active transport typically is used for larger polar molecules that you're trying to transport against a certain concentration gradient. So for instance, if you're trying to move sodium, let's say from inside of the cell to outside of the cell, but the concentration of sodium is higher outside of the cell, then it's going to take some energy to move that sodium against its concentration gradient to an area of higher concentration outside of the cell. That would require ATP and therefore would be active transport. Now there's different types of active transport pumps. We have the sodium-potassium pump and calcium pumps and different types of transporters as well. The sodium-potassium pumps you find in every single cell in your body, and really what these sodium-potassium pumps do is they use ATP to pump 3 sodium out and 2 potassium into the cell. Okay, and what this does is it helps establish a concentration gradient that can be used for things like action potentials or even transport of molecules into and out of the cell. Now, we also find most cells have these membrane calcium transporters. Because calcium is an important intracellular signaling atom, we need to make sure that it's transported and stored in the appropriate areas. So these membrane calcium transporters can basically use ATP to pump calcium into a certain area. Like in muscle cells, a membrane calcium transporter will pump calcium into the sarcoplasmic reticulum. Or in other cells, membrane calcium transporters can pump calcium out of the cell and keep calcium concentrations low inside of the cell. Now, other membrane transport carriers could be like sodium-driven carriers and passive transport carriers. A sodium-driven carrier basically can use the movement of sodium to help transport another molecule. Like for instance, you could actually have sodium move into the cell, and the movement of sodium into the cell can also help bring glucose with it. We call those sodium glucose transporters, and that would be an example of a sodium-driven carrier. But another example would be like a passive transport carrier, where basically it's just just a route for a molecule to get into out of the cell, but doesn't require the use of ATP. That's just showing the sodium-potassium pump, where we can use ATP to pump 3 sodiums out and 2 potassiums in. And it turns out that the activity of the sodium-potassium pump really helps keep the cell alive in a lot of different ways. We'll talk a lot about what happens when sodium-potassium pumps fail in dying cells in like future chapters. And it'll make more sense when we come back to this in a little bit. Now, this is an example of just using ATP to bring something into the cell. So this would be an example of an active transporter, where we actually can take ATP, use the energy of ATP to literally pump an atom or molecule into or out of the cell. And that's just this example here. Now, passive transport can actually involve just these little passive transport carriers here. So let's say we wanted to bring glucose into our cells. Well, all that requires then is just to use this little membrane carrier. And as long as glucose is in higher concentration outside the cell and lower concentration inside the cell, glucose can get into the cell easily just down its diffusion gradient. Now, these protein carriers, though, aren't always in the plasma membrane. And really what helps bring those to the membrane is when insulin binds to its receptor, that can activate this intracellular signaling cascade that allows for these transporters to move to the plasma membrane. And once in the plasma membrane, these transporters can help bring glucose into the cell. And so that's one of the effects of insulin here. Now, these don't require ATP. So basically what you're looking at is just for glucose to bind to this transporter, it gets basically shuttled into the cell through this transport protein. No additional ATP required, so we call this passive transport. Now, there's lots of different ways that cells can communicate with each other. We have gap junctions and direct contact, as well as secretion of chemical mediators. Remember, gap junctions are basically little protein tubes that allow for molecules and current to directly spread from one cell to the next. You're going to find these in like cardiac muscle, as well as many developing cells. There are other mechanisms where cells can communicate through direct contact, where cells can bind to other cells, and that can initiate communication between the two cells. Otherwise, most signaling mechanisms between cells occur via secretion mechanisms, where basically cells can secrete like a hormone or a short distance sort of chemical mediator that can communicate with cells in like a short distance, like a synapse, or long distance, like a hormone. So what we see here is like sometimes signaling molecules can be released by a cell, and those can travel throughout the body and bind to receptors in a target cell. Otherwise, we can have direct contact signaling, where one cell can bind to another, and that can initiate communication between the two cells. Or a cell can bind to like the matrix or like its protein scaffolding in a tissue, and that itself can be the communication for a certain reaction. Like, for instance, like when blood clot forms, that's due to the communication of certain blood cells with the exposed matrix of that damaged blood vessel. And this communication between the two components helps initiate a blood clot. Now, this differs from gap junctions, which are basically a little protein tube that connect cell to cell, and so currents can spread from one cell to the next just through those protein tubes. And you're going to find this in areas like your heart, where electrical currents can spread from one cardiac muscle cell to the next cardiac muscle cell. Now, other types of signaling molecules or signaling mechanisms can include things like neurons, which can actually communicate with other target cells at a synapse, where neurotransmitters in vesicles can be exocytosed, and those neurotransmitters can bind to receptors in a target cell. This differs from like paracrine signaling, because paracrine signaling occurs when cells in a given tissue communicate with other nearby cells in that same tissue. We call this paracrine signaling. Now, paracrine signaling differs from endocrine signaling because endocrine signaling requires the long-distance communication of cells throughout the body, usually requiring your bloodstream or the lymphatic system, which can help transport hormones that are secreted by a cell throughout the body. Those hormones will diffuse out of the bloodstream or lymph and then basically bind to a receptor on a target cell. Now, autocrine signaling is when a cell actually excites itself. Usually, autocrine signaling is involved with development as well as sort of regulation of how much secretion of a hormone or neurotransmitter is occurring. And we call this autocrine signaling, where a cell is basically signaling itself or releasing a signaling molecule that binds to a receptor on the same cell that released it. Now, what happens is that not all receptors are the same. We have ion channel receptors and enzyme-linked receptors as well as G-protein-linked receptors. Now, ion channel-linked receptors are basically receptors that, once activated, open up an ion channel and allow for ions to flow, like let's say sodium flowing into the cell, or potassium flowing out of the cell, or calcium flowing into the cell. Basically, this receptor is also an ion channel, and so when the signaling molecule binds the receptor, it's going to open up that channel and allow for basically ions to flow through. Now, this differs from enzyme-linked receptors because these enzyme-linked receptors basically are receptors that, once activated, they can activate a more complex intracellular enzyme cascade within the cell, and those enzymes can do a lot of different things in the cell. They can change cellular metabolism, they can change gene expression, they can turn on or turn off different cellular processes because these enzymes basically can have a variety of functions depending on the type of receptor. So we'll talk about MAP kinase pathways and JAK-STAT pathways, but basically these are just enzyme-linked receptors that are associated with certain processes. Now, G-protein-linked receptors can also change cellular metabolism, and these are more associated with things like glucagon, epinephrine, and vasopressin, and other hormones. These G-protein-linked receptors are important pharmacologically because half of all pharmaceutical drugs target some sort of G-protein-linked receptor. So they're very diverse types of receptors that can have a variety of intracellular effects, and they're very modifiable and changeable. So it's hard to predict what exactly a G-protein-linked receptor will do to the cell because it can have a variety of effects. But we do know that it actually can change the metabolism or change the cellular processes within a given cell. Now, these G-protein coupled receptors are pretty complex, and what this slide is showing is basically this process where this is the G-protein coupled receptor here, and once activated, like glucagon, epinephrine, or vasopressin can bind to the receptor and then activate this G-protein coupled cascade, which activates other enzymes like protein kinase, and those can go on and turn off different enzymes in the cell, which can change things like gene expression. So just one thing to kind of take away from this slide is that G-protein coupled receptors can change gene expression in the cell, which can change the metabolism of a cell. So it's a pretty interesting type of receptor. Now, this differs from something like a tyrosine kinase receptor, which would be something that's associated with like insulin, where it's still an enzyme-linked receptor that once our hormone binds the receptor, it activates these enzymes that can activate other enzymes that can change the metabolism of the cell. Again, because it's a more complex pathway, there's lots of potential pharmaceutical targets for aspects of this type of receptor. Now, we're going to wrap up this chapter talking about how cells can grow and divide. Now, after we've been fully developed, cells need to be kind of more strictly controlled in terms of how they're able to grow and divide. Now, what's important then is that once someone is fully developed, the rate of cell growth should match the rate of cell death. And if there's a disparity between the two, then that's when you can get problems. Like if you have more cellular growth than death, that's when you can get tumor formation. Or if you have more cellular death than growth, that's when tissues and organ systems start to break down because cells aren't being replenished fast enough. Now there's two main types of cell division we'll talk about. That's mitosis and meiosis. Mitosis is the type of cell division that most of the cells in your body undergo. And from mitosis, one cell can divide into two, and mitosis yields two daughter cells that have a complete set of chromosomes. Now this differs from meiosis, where meiosis only occurs in germ cells, or basically sex cells. And so we're talking about like sperm and egg, how they're formed. Now meiosis is different from mitosis because from one cell undergoing meiosis, we have two rounds of cell division. So from one cell, we actually get 4 germ cells that can result. Now what's interesting then is that in meiosis, we still have a complete set of chromosomes, but we have half of the number of copies of those chromosomes. So the cells that result from meiosis we talk about as being haploid, or have half of the number of normal chromosomes, like half the number of copies. They have a full set, but only half the copies. Now if you think about it from a standpoint of sort of reproduction, if the sperm brings in half the number of copies and the egg has the other half of copies, then those together can make a full set of copies of chromosome for that new zygote. So when you think mitosis, think most cells in your body dividing from one cell into two. And meiosis is where from one cell, typically you're going to get at least 4 sort of daughter cells that can result. Now, in cellular division, we have some key phases here. We have our M phase, our G1 phase, S phase, and G2. Now, M phase is really when the cell is undergoing division itself. Okay, so M phase, as we call is the mitosis phase, when the cell is actually dividing. Now, G1, S phase, and G2 are all phases of what we call interphase. And interphase is all of this outer ring right here, not including M phase, that the cell mostly spends its time within. So when you think about a cell that's not currently dividing, it's an interphase. And within that, we'll have either G1, S phase, or G2. Now, G1 is a phase of interphase where right after cell division, the cell can kind of check to make sure that everything divided properly. And before it's able to move forward in other interphases, what we have then is this thing called a restriction point, where the cell can be kind of halted and prevented from further developing. And this restriction point is important for cancer because as long as you have a restriction point, these cells won't divide very rapidly. They'll divide more slowly. But if the restriction point doesn't exist, these cells will divide extremely rapidly, and that's where you can get tumors and possibly even cancer that can result. Now, from G1, we can enter the S phase, and during S phase, this is called the synthesis phase, where basically the cell is actually dividing its chromosomes, it's dividing its organelles, and preparing for cellular division. Now, if we continue from S phase into G2, G2 is basically kind of like a checkpoint phase, where basically the cell has mechanisms that allow it to check and make sure that all of its chromosomes are replicated and all of its organelles are replicated. That way, when the cell undergoes cell division, the daughter cells will have copies of chromosomes and organelles. You know, if you didn't have copies of your mitochondria and your cell still underwent division, then there's a good probability that one cell might not have any mitochondria at all, and it's not going to be a viable cell at that point. So it's important then that the cell can check to make sure that all of its organelles and chromosomes are copied. That way, before going into M phase, where the cell starts to divide, we can make sure that each cell has, you know, a set of copies of each thing that require it to survive. Now, there's several key phases of mitosis. We have prophase, prometaphase, metaphase, anaphase, telophase. And then what's really not mitosis, but it's called cytokinesis. That's just where the cell kind of starts to split apart. Now, basically mitosis starts with prophase, and prophase occurs when the chromatin starts to condense into actual chromosomes and the nuclear membrane starts to break down. Now, the prometaphase is when the mitotic spindle starts to develop and chromosomes start to be pulled on this mitotic spindle. Metaphase is characteristic when the chromosomes line up at the metaphase plate in a long line here, and they're preparing for basically chromosomal separation, which occurs during anaphase when the sister chromatids of each chromosome get pulled apart to opposite ends of the cell. Basically, each sister chromatid is like a copy of the entire chromosome. So up here we have 2 copies of each chromosome, and down here each copy is being split to the opposite side of the cell. So this copy of the chromosome is going to this side, this copy is going to this side. You can see that each chromosome has a copy going to opposite ends, and they're being pulled apart by the microtubule spindles of the mitotic spindle, which is bringing these chromosomes to the opposite ends of the cell, which will later become what appears to be like 2 different cells. This is called telophase, when all those little copies of chromosomes get repackaged into little nuclei, and the cell starts preparing for division. And once a cell actually starts splitting into 2 separate cells, we call that cytokinesis, which basically means the movement of cells.